Good afternoon, students. Today we'll start our lesson with a recapitulation of what we did yesterday. I hope you understood yesterday's lecture. So we will start with the monohybrid cross which we discussed yesterday. Now here you see what is on your displayed on your screens. Yesterday we dealt with the tall and the dwarf one, so I thought I'll take another example today, that is the round and the wrinkled seeds. Now if we see, you see the parental generation. This is just a recapitulation, so I will just brush you up and we'll hurry up. So if we see, this is the round seed, this is the round seed, right here. Again, one factor here is round. So you see in the parental generation here, what you see, the female, though two different crosses are displayed on your screens. One is showing the female as the round and other is showing male as the round parent, right? So if we take the cross 1, then round I have marked with green and wrinkled I have marked with red, right? So here it is the wrinkled seed and here is the female is the wrinkled parent. According to Mendel's law, in the first filial generation, that is the F1, all the plants were found were what type? Round type, no red type. And even in the other cross, all were round. This means irrespective of the parent, whether male or female is having the round character, the net result was that round was found in the F1 generation in all the offsprings, or you can say in all the members of F1 generation. Then selfing of the F1 generation takes place. You know, there is cross fertilization only once, that is amongst the parents. After that, in the F1 generation, in order to reach the F2 generation, selfing is done of the F1 generation. That means self-fertilization. So as a result, what we observe is 3. Here you see 3 parts of it are green, that is the round ones. Again, irrespective of the fact that whether the male is round or the female is round, but a round character is expressing itself. So we can say that the round character is a dominant character. The character which expresses itself in the F1 generation is the dominant character. Why? Because it is not allowing the other gene to express itself, that is the other allele of that gene or the other factor in Mendelian inheritance or Mendel's terms, the factor, only the dominant factor is going to express itself. So in both the crosses you can see that in the F1 only round character is expressed. Okay. So up till here it's clear. And in the F2 you see, now we don't have all round. Again there is appearance of the wrinkled one. You know, we know it is round, so we will call it dominant. That is not the thing. The thing is the character which is expressing itself in the F1 generation. That is why we are calling round as the dominant. Had wrinkled expressed itself, we would have called wrinkled as dominant. But the fact is, the experimental proof is that round is dominant. Round is expressing itself. So round seeds are dominant character. So in the F2 you see 1/4 part is red, rest 3/4 is green. Now we have already discussed this yesterday, that out of this 1/4 part is the homozygous and 2 parts is the what? Half of it is what? The hybrid ones, right? So this is analyzed again. How can we analyze this? Because phenotypically you see it's the round seed, right? So F2 में round आ गया. Parents हमने start किया एक round एक wrinkled. कैरेक्टरिस्टिक फीचर है ये इनका, वी आर फोकसिंग ओनली ऑन द वन कैरेक्टर। ये हमने अर्जुन की आंख वाला फिनोमिनन लिया है कि मछली तो पूरी मूव करती है लेकिन उसकी आंख पे उसका फोकस था। सिमिलरली हम यहाँ पर सिर्फ एक ही कैरेक्टर के ऊपर फोकस करेंगे, दैट इज़ द शेप ऑफ द सीड, राउंड और रिंकल्ड। बाकी पूरे प्लांट की तरफ हम नहीं देख रहे वो किस तरह ग्रो कर रहा है, राइट। सो F1 में राउंड, F2 में अगेन अपीयर हो गया रिंकल्ड, 1/4 पार्ट। अब हमने फिर से सेल्फिंग करके F3 जनरेशन पे जाएंगे। कल हमने F3 नहीं डिस्कस किया था। सो F3 में क्या हो जाएगा, दिस इज़ द थर्ड जनरेशन, थर्ड फीलियल जनरेशन। इसमें सब सेल्फिंग कर रहे हैं, ये जितने भी टाइप्स हैं उन सब की सेल्फिंग हो रही है। सो सेल्फिंग में क्या होगा, सेल्फिंग का रिजल्ट अब देखिए आपके आगे डिस्प्लेड है कि सेल्फिंग का रिजल्ट क्या है। सेल्फिंग का रिजल्ट ये है कि जो तो रिंकल्ड सीड था वो तो प्योर है अपने कैरेक्टर के लिए, वो हमेशा ये सारा का सारा जो है ये यू सी all the four they are red. Excuse me. So all the four they are red, whereas here you see in these middle two rows you see one part is red, but then rest all are green. This, they are all green, right? Now what do you deduce from here, इससे क्या रिजल्ट आपको मिलता है। F2 में जो होमोजाइगस था वो ट्रू ब्रीड कर रहा है, सारे के सारे जो हैं वो राउंड सीड्स हैं बिकॉज वो होमोजाइगस है। सिमिलरली जो हेट्रोजाइगस थे राउंड, उनमें ये रिंकल्ड करेक्टर दोबारा अपीयर हो गया है। ये रिंकल्ड करेक्टर उनमें 1/4 अगेन F2 की तरह उसमें फिर से अपीयर हो गया क्योंकि वो F1 की तरह ही हाइब्रिड्स हैं, राइट। और फिर यहाँ पर सिमिलरली दोनों क्रॉसेस में सेम रिजल्ट्स हैं। सो इररिस्पेक्टिव ऑफ द मेल एंड द फीमेल पेरेंट, कि कौन सा पेरेंट मेल है कौन सा फीमेल है, इररिस्पेक्टिव ऑफ दैट यू सी द रिजल्ट्स आर सेम। I hope you understood this now by now. Shall we proceed further now? So we'll see now here, you see that we will move now to the next slide, that is the what we are going to talk about today is कोडोमिनेंस। ठीक है, मैंने आपको ये पहले थोड़ा सा ब्रश अप कर दिया ताकि आपको ये फैक्ट्स क्लियर हो जाएं, एक बार में शायद आप नहीं उनको रिमेंबर कर पाओ। सो वी विल स्टार्ट नाउ विद कोडोमिनेंस। व्हाट डू यू सी डिस्प्लेड ऑन योर स्क्रीन्स? Yes, you see two examples displayed for codominance, right? Now if in the chick you see there are two different colors in the parent and you see both the colors they are expressed in the progeny. Similarly in the cow also you see this red and the white colored cow, and here the progeny is having patches of red and white, that is the roan. So what do you understand here? अभी तक हमने क्या पढ़ा था? डायरेक्ट डोमिनेंस एंड रिसेसिव। या तो वो कैरेक्टर एक्सप्रेस हो रहा है या नहीं हो रहा है, डोमिनेंट रिसेसिव। अब आप देखोगे कि ये कोडोमिनेंस मीन्स कि दोनों अपने आप को एक्सप्रेस करेंगे, कंप्लीटली रिसेसिव नहीं है कोई। वो नहीं कहेगा कि मैं रिसेसिव हूँ, वो कहेगा भाई मैं भी हूँ, मेरे को भी अपने आप को एक्सप्रेस करना है। So both, when both the alleles they express themselves in the hybrid, then that is what is known as codominance. ठीक है, दोनों ही डोमिनेंस हैं बराबर के। अब इसका एग्जाम्पल हम स्टडी करेंगे। सो वी कम डाउन टू codominance. So what is codominance? When two alleles they express themselves together, when both of them they express themselves together, that is what we call it as codominance. Like you see in the flower color, here you see both the colors, the red and the white, they are expressed in this flower. Or you see the blood types. Blood type is the best example taken for codominance. It is even taken for multiple allelism, which we'll take a little later on. So blood types, the human blood types. We have blood type A, blood type B, blood type AB. So blood type A has cells with A proteins, B has cells with B proteins, but blood cell AB has blood cells with A and B proteins. So here you see A type, B type, and in the center is AB type, in which both are present, A as well as B, they are both present. So here you see this is the A type and this one is the B type, whereas here you have both A as well as B type. So this is what is codominance. I hope it is clear to you. So we'll move further with codominance only. So we'll take here this example of codominance, that is the example of the blood group. So codominance is when the heterozygote shows the traits associated with both homozygous genotypes. So you see this codominance in molecular traits, and the most common type of codominance is associated with the blood types. Here there are multiple alleles that determine the blood type. That is more than 2 alleles when they are present for a particular gene, that is known as multiple alleles. I'll discuss this in detail a little after discussing the codominance. So coming back to the blood type. Here you see the blood type and its genotypic and the phenotypic, what it is. Right, so the blood type A, it has genes we take as the, let me tell you one more thing. Now first let me tell you what these alleles they represent. So now, as I told you a little before only, that we were discussing yesterday and even today in the start about the F1 which resembled either of the parent, that is either dominant or recessive. Or yesterday we even discussed the incomplete dominance, that is a character which was present in between the two. Antirrhinum majus, right? Snapdragon. Now, but when we talk about codominance, here the F1 generation resembles both parents. A good example is different types of the RBCs that determine this ABO blood grouping in humans, which we are going to discuss just now. Now, this ABO blood groups are controlled by the gene, let's say, take it as a capital I gene, right? So we talk about the blood gene as the capital I gene, right? Capital I. Now the plasma membrane of the RBCs, they have sugar polymers that protrude from its surface, and the kind of sugar is controlled by this gene. So the gene I, it has 3 alleles. It can have the allele capital I raised to the power, this you can write here as IA, as displayed on your screens, or you can write it as, this is the IA, or we can write it as for B blood group as IB, right? So the gene I has IA for A blood group, you can say IB, and then there is this, either we write it as I raised to the power O, or we can also write it as small I, right? So we also write it as small I. So in some books you will find that this is written as small I. So alleles capital I A and capital I with a superscript B produces a slightly different form of the sugar, while the allele small I or capital I with superscript of O does not produce any sugar. But humans are diploid organisms, so each person possesses any two of these three genes alleles, right? So any human being will have only two of these three. These are three alleles of the same gene I. So any two are present in one human being. एक इंसान में दो ही present होंगे इन तीनों में से, right? So whenever this gene IA and IO, this one is present, or only this IA and IA both the alleles, same alleles are present, then the blood group will be A type and the genotype will be AO or capital A and capital A. So when we have this I gene with superscript of B along with I gene and superscript of O, then the blood group will be B type and the genotype will be BO. We can also talk about it as if they are same alleles, that is I superscript B and I superscript B, then again the blood group will be B. Because both the genotype they are having B, so this is homozygous B. Blood group is same, but it is either heterozygous B or homozygous. Same is the case for A blood group. It can be homozygous A or heterozygous A. Okay. But when both these alleles are present, supposing one parent has this capital I gene with superscript of A and the other one with B, then the blood group will be AB type. Both are equally expressing themselves, so the blood group becomes AB, right? So they are present together and they both express their own types of sugars. This is because of codominance. Clear? So here the RBCs, they will have both A and B type of sugars, but there are 3 different alleles. So there are 6 different combinations that these 3 alleles they make possible. And the last one is the possibility of the O blood group. That means when neither A nor the B carrying allele is present. Both are this same. That is for O blood group. Small I you can say, or you can say that they are both this not having either I, not A nor B allele. So the blood group here becomes O. So now from here you understand how many genotypes and how many phenotypes we have. So how many phenotypes are possible? Can you tell me? Just see. Phenotype A, phenotype B, phenotype AB blood group and O blood group. This means there are 4 types of phenotypes, but genotypically we have 6 types. That is capital AO, then capital A, then capital BO, B and B, then AB blood group and O blood group. So genotypically we have 6 different possibilities, whereas phenotypically we have only 4 possibilities. Clear? I hope so. In case of any doubts, you can ask me, you can ping me. So now we'll take the this example to illustrate it further, to make it more clear to you. So as I told you, the genotype carrying both the 8 alleles or one A allele will have this type of appearance as shown on your screens, and the phenotypic blood will be A. Similarly, in the next type, when both the alleles are carrying the B type sugar or only one B type, then you can see the structure of the sugar in the appearance, and the blood group will be B type. The third case is when both are present. When both are present, then you see the structure of the sugar RBC, and then the blood group becomes AB. And the last case, you see nothing is present on this RBC, and that is O type. So here you see all the 4 types of phenotypic expressions of the blood group and how they are possible. Okay. So now do you understand how we get our blood groups? So in codominance, both the alleles, they are expressed in the phenotype. Here you can see on your screens, the father is having A blood group, whereas the mother is having B blood group. Their child has blood group AB. Is it clear how AB? So I hope now you can solve the problems related to blood group. I am going to give you some. So how this multiple allelism, the term I told you in the starting of codominance. So codominance and multiple alleles. So the traits involve only 2 alleles per gene. You saw up to incomplete dominance in the monohybrid cross, in incomplete dominance, right? But for most of the genes, more than 2 alleles also exist. For example, what we have discussed just now, that is blood types in humans. Here a person can have how many type of blood groups? 4 types: A, B, AB and O. We just discussed. So each letter refers to 2 types of carbohydrates, that is A for A and B for B. So now can you tell me why it is so essential, so critical to match the blood types? Can you tell me? Think over it. Yes, to avoid agglutination of the blood, that is to avoid blood clotting. If A blood is given to a person having B blood group, the blood will clot, or vice versa. That is why it is very difficult or very essential that whenever there is blood transfusion, blood matching has to be done. And let me tell you one more thing, blood matching is done not only for the blood group, but it is also done for the rhesus positive or the negative aspect, that is positive blood groups and the negative blood groups. So now children, once again telling you something about multiple alleles. So when instead of having 2 forms of an allele, there are more than 2 forms for one gene, that is what is known as multiple alleles, and the phenomenon is known as multiple allelism. And the best example for humans is the human blood. So we have here 3 types of alleles, that is A, B and O. Now here the A and the B alleles, they are codominant whereas the O one is recessive. Okay. So according to multiple alleles, if we talk about the blood types, again we are discussing the same example, that is the blood typing, but with a different view, that is view of the multiple alleles. So here you can see that the blood typing alleles, this having I to superscript as A or B, they are both codominant, whereas the third allele is recessive. So blood typing genotypes you see here, they are the same what we discussed just now, but here we are discussing it in this form. The blood typing genotypes and their phenotypes, this small I will yield the type O, whereas A and B, both alleles, if they are present, they will yield the blood type AB, and homozygous or heterozygous A will give type A, whereas the homozygous or heterozygous B will give that blood group B type. So here our view is of the alleles. The example is same, blood groups. First we discussed it with dominance, that is what type of dominance, recessiveness it shows. So it was a codominant example. Now the number of alleles which are present, more than 2 alleles, that is 3 alleles are present here, and we call it as the multiple allelism, and we are discussing the same example with respect to the number of alleles, that is the multiple alleles. So what are the features of the multiple alleles? So multiple alleles always occupy the same locus on the chromosome. You know, Mendel talked about factors and today we know about the chromosomes, about genes, about the loci or the locus on the genes. So multiple alleles, they always influence the same character. No crossing over among the member alleles of the same multiple allelic series occurs. So these are some important points to remember about the multiple alleles. I'll repeat them again. Multiple alleles always occupy the same locus on the chromosome. Multiple alleles always influence the same character. No crossing over among the member alleles of the same multiple allelic series. Now let's see what this law of segregation tells us. Law of segregation. So traits are controlled by particulate factors which are always present in pairs and get separated during the gamete formation. Here you see in a cell having 2n number of chromosomes, the homozygous chromosomes, they have been marked with different colors to show the same character or the same gene. After meiosis 1, the homologues, they separate, and this is known as the reductional division. Then that means in the 2n or the diploid cell, one chromosome was from the mother and other was from the father. After meiosis 1, they have separated out. Now in meiosis 2, that is again an equational division, you see 4 gametes are formed having n number of chromosomes, of which 2 have one type of character and the other have second type of the character. You can see 2 with green color and 2 with black color. Okay, clear? This is how they segregate. So traits, they are controlled by particulate factors which are always present in pairs, and they get separated during the gamete formation. I hope things are clear to you. Now we come down to the inheritance of two genes. Up till now, whatever we were discussing, we were taking only one gene in consideration. So when we talk about two genes, we talk of the dihybrid cross. When we talk about dihybrid cross, we talk about Mendel's dihybrid cross. So dihybrid cross is a cross between two different lines or genes that differ in two observed traits. According to Mendel's statement, between the alleles of both the loci, there is a relationship of completely dominant recessive traits. Right? So now we are going to discuss about the dihybrid cross, which is now displayed on your screens. So again we start with the parental generation. Okay, but you see here that the genes into consideration are now two characters are being taken into consideration. So when we talk about the parental generation, we are talking about, our focus is on two characters. Here the color and the shape of the seed. One of the parent having yellow color and round seeds, whereas the other having the green and the wrinkled one. Here you can see on your screens, one parent is round and the other is green and wrinkled, right? So two characters you see here, round and yellow, wrinkled and green. Now keep one thing in mind, that they are pure lines. Whenever we take the parental generation, we take pure lines, and this pure line is true for both the parents. The round and yellow one will breed true to this character in all the offsprings, and wrinkled and green will also be true to its traits. Now either can be male or female, this we have already discussed, it does not matter. Now the gametes which are formed, now these are the gametes, both these. So when gametes are formed, I just told you that from the true only one will pass on, that is from 2n number the gametes have the n number. So chromosomes from 2n will come down to n number. Reductional division will be reduced to 1/2. So only one of the representatives, that is one y of the 2 is written here, and one r of the 2 is written here, that is the capital Y and capital R for round and yellow, whereas for green and wrinkled the small y and small r. So now you know why we write two y's and two r's in the parents and one y and one r in the gamete. So these gametes, they are cross fertilized. They are cross fertilized. So as a result of cross fertilization, you can see the resultant F1 generation. The resultant F1 generation is here having what? The yellow and round. So all the progeny of F1, they are having yellow and round seeds, but They are heterozygous, mind you. They are heterozygous. They have one capital Y, one small y, one capital R, one small r. Okay. So they are heterozygotes. But in phenotype, they are all like the parent having the round and yellow seeds. They resemble that parent, only one parent. So F1 progeny is completely showing the dominant recessive characteristic, right? So this characteristic is then studied further in the next generation. I hope you remember that while we are studying these crosses, there is a long process of first generating pure lines of the parents, then growing them, then artificial hybridization, that is for F1, and then again producing the seeds of the F1 and sowing them in the next generation, and by self fertilization or selfing they will show the resultant F2 generation. Now here in the box you can see the gametes are shown here outside. So how many types of gametes are formed? Capital R, capital Y, capital R, capital Y, small y, capital R, small y and small r. These we can take as gametes of one parent, let's say male parent. Similarly for female parent, capital R, capital Y, small y, capital R, small y and small r. So how do we deduce what is formed? This is shown on a piece of paper, but actually such seeds are produced as a progeny. This F2 progeny, the second filial generation. So F2 progeny, how do we represent them? You see here that the F2 you can represent like you see this capital Y, capital R, capital Y, capital R of male and female, they will form this progeny showing the homozygous character for both the yellow and the round seeds. Similarly, you can see here the next one, that is the capital Y and the small y, capital R and small r here, right? So this way you will follow this whole line with capital Y and capital R with these. This will be formed like this, with this we will calculate like this. Okay. Similarly, the next row will be calculated with capital Y and small r along with again these like this. So here we will get the next progeny. That is, if you look at the second row, it is capital Y, then capital Y, capital R and small r. Then in the next one you see capital Y, then small y and then small r and small r, right? So capital Y, small r and small r. Then capital Y, small y, capital R and small r in the third one here. Similarly this last one. So here you see in the last block we find small y, small r here along with small y and small r of this side. So you can make this whole block by looking at these combinations. Now this combination gives us a result showing a particular phenotypic ratio which is known as which ratio? It is the 9:3:3:1. It is 9:3:3. I will just show it here. 9:3:3:1. So this is the ratio of a typical dihybrid cross, Mendelian dihybrid cross, which shows complete dominance and recessiveness. So this shows that there is a combination series of 3 yellow is to 1 green with 3 round is to 1 wrinkled. This derivation we can write, with different, showing this phenotype as 9 types which are round and yellow, 3 which are wrinkled yellow, 3 which are round and green, and 1 which is completely recessive, that is wrinkled and green, which resembles the parent one. And the first one also resembled one of the parents when we started it. So with this Puneet square we can effectively understand the independent segregation of two pairs of genes during the meiosis and the production of the eggs and pollen in the F1, F2 generation, right? So now we see that all the 4 types of gametes, now you understand that there are 4 types of gametes formed when we start with the F2 generation. For F2 there are 4 different types of gametes of male and the female. So this means the frequency is 25%. So now is it clear? Okay. So now you can see this. I'll repeat again. We talk about the parental plants. Then we come down to the gamete formation, one type of gamete. Then the one type of F1 progeny. F1 progeny forms 4 types of gametes with a frequency of 25% for each type, and their self-fertilization leads to the formation of a progeny with a phenotypic ratio of 9:3:3:1, right? So this is what I am telling you. 9 are round and yellow, 3 are round and green, 3 are wrinkled yellow, and 1 is wrinkled green. So you see maximum here also they are round and yellow, because even if one of the 2 dominant alleles is present, it will show the dominant character. You see, out of all these, only this last one is showing 1 is wrinkled and green, because no dominant allele is present in it. Now I am giving you one small assignment, that is you calculate the genotypic ratio of this cross. Okay. So we come to the law of independent assortment. So the Mendel's law of independent assortment states that the alleles of 2 or more different genes get sorted into gametes independently of one another. In other words, the alleles of a gamete receives for one gene does not influence the allele received for another gene. So the assortment is independent of one another. So I hope you understood it, students. So we'll again meet on Friday. Till then, stay safe and stay healthy.